Modern agriculture is on the brink of a new era, one that represents a quantum leap into the 21st century. Although there will always be a need for conventional agricultural methods, there's no doubt that the green revolution of a quarter century ago is rapidly giving way to today's gene revolution. The cornerstone of this new biotechnology is the propagation method known as tissue culture. Tissue culture is a relatively new laboratory science which encompasses a variety of procedures and techniques. Simply stated, tissue culture is the method by which plant and animal cells and tissues are grown on a sterile media. Until recently, most tissue culture studies were directed at medical and drug research. But now, scientists are broadening their perspectives to utilize tissue culture in nearly all phases of agriculture with tremendous possibilities. For example, animal scientists use tissue culture to accomplish embryo transfer and bovine embryo division. Forestry professionals are using tissue culture to significantly increase productivity of Douglas fir and loblolly pine plantations, and to quickly propagate superior specimens of slow-growing trees. In the food industry, tissue culture provides a means by which protein-rich sweeteners are being developed for baked goods, jams, ice cream, and candies to raise the nutritional value of these foods. Aided by tissue culture, plant pathologists have created experimental bacteria that inhibit frost formation on plant leaves. Soon, strawberries and other tender crops will be able to sustain temporary periods of low temperatures without damage. Crop scientists have made spectacular advancements through tissue culture. Researchers are working on tomatoes that withstand herbicide sprays and thrive in salty soils, corn hybrids capable of producing their own nitrogen fertilizer, and super grains that tolerate extended drought. Currently, tissue culture is more widely used in commercial horticulture than in any other area of agriculture. It was the orchid industry, more than 25 years ago, that first used tissue culture on a large scale. Orchids are notoriously slow to reproduce, and their seeds produce variable plants. To compound the problem, orchids are extremely susceptible to virus infections that are easily passed through conventional propagation methods. However, by surface sterilizing the virus-free shoot tips of orchids and propagating them by tissue culture, growers are assured that the resulting plants are free of pathogens and identical to the original plant. Nursery professionals are finding that tissue culture produces these genetically identical plants quickly, economically, and with less labor, space, and maintenance than is needed by traditional methods. For example, one grower uses tissue culture to produce 1,000 daylilies each week on just 30 square feet of shelf space. Another grower, using conventional propagation methods, would need a half acre to produce the same number of daylilies. Tissue culture techniques also enable growers to produce vast numbers of plants when mother stock is in short supply. As another example, Dutch iris normally produce about 5 daughter bulbs per year under optimum conditions. But with tissue culture, you could produce several hundred Dutch iris from a single bulb within 3 months. The potential for rapid mass production makes tissue culture particularly appealing in commercial horticulture. An Oregon nursery produces 5 million certified tree root stocks each year, while a foliage plant nursery produces 100,000 Boston ferns each month. One of the largest tissue culture labs in California plans to produce more than 25 million plants annually. Because tissue-cultured plants have a guaranteed health status, it's possible to transport plants into countries formerly inaccessible due to strict quarantine regulations. This means we can look forward to rapid worldwide introduction of new and improved plants in the not-too-distant future. On the research level, horticulturists are using tissue culture to create new plants from totally unrelated families. They're also able to alter the number of chromosomes in plants. They can breed plants with improved form, vigor, and productivity. Techniques such as gene transfer that were once regarded as science fiction have become reality. By fusing into one cell the contents of two genetically different cells, researchers hope to produce a blue rose, a blue chrysanthemum, and flower colors coordinated to the season's changing fashions. Another new technique is called embryo rescue. Plants that fail to produce viable seed can be tissue cultured by this method before the fertilized ovary, or immature seed, degrades. Embryo rescue is a valuable tool in the production of new lilies. With mutagenesis, scientists can induce mutations in an effort to develop plants resistant to disease, cold, toxins, and herbicides. Tissue culture also enables long and short-term germplasm storage. With this method, the genetic material of old and rare plants can be preserved from extinction. In production, a rhododendron nursery uses germplasm storage to hold its weekly output of over 1,000 plantlets until the proper planting season. Rounding out the newer tissue culture techniques in horticulture is encapsulation. This involves the formation of embryos from single identical cells in the laboratory. The artificial embryos are coated with a gel that may contain fertilizers, herbicides, pesticides, growth regulators, and fungicides. The resulting seeds produce identical plants, giving uniform results at low cost. Potentially any living cell in the tissue of any plant can grow into a normal plant through tissue culture. Because such small pieces of plant tissue are used in tissue culture, the procedure is often referred to as a type of micropropagation. It's equally well known as in vitro propagation, a Latin term meaning in glass. In the beginning, all of the plants produced by tissue culture were grown in glass test tubes, jars, or petri dishes, but today modern plastics are used as well. The tissue obtained from the mother plant is called an eggplant. The resulting offspring are called clones because they're genetically identical to the parent. We've seen that tissue culture is having an important impact in the field of agriculture. We've seen several important advantages of production with tissue culture. These include improved propagation efficiency, the development of pathogen-free planting stock, and the capability of storing germplasm for later use. Propagation by tissue culture also produces plants that are the exact replicates of the mother stock. Now let's turn our attention to a laboratory's basic layout and the equipment used in propagating plants by tissue culture. Tissue culture labs vary in size and sophistication. However, all labs share several basic features, whether large or small. It's most important that aseptic working conditions are provided. That is, the laboratory must be free of bacteria, fungi, and other microorganisms. All working surfaces, tools, and equipment are regularly sterilized and kept free of contamination that would hinder plant growth. Typically, a tissue culture lab consists of 3 workspaces: a preparation area, a transfer room, and a growth room. The preparation area is the scientist's kitchen, where plants, equipment, and the sterile nutrient media are readied for culturing. Look around and you'll find the basic tools and equipment used in the preparation area: a refrigerator to store the chemicals, a precision scale for accurate measuring of ingredients, a hot plate for heating media, a pH meter to measure the alkalinity or acidity of the nutrient media, and an autoclave to sterilize tools and glassware. An autoclave is a pressurized steam sterilizer commonly used in commercial labs. To save money, some smaller labs often use a pressure cooker for sterilizing tools and glassware. Through a restricted passage which minimizes the entry of airborne microorganisms, we find the transfer room. This is where the cultures are started, divided, trimmed, and moved from one container to another. In this room, laminar airflow hoods, called transfer chambers, continuously circulate clean air to remove dust particles and to provide a sterile working environment. A dissecting microscope may be used to aid in the accurate division of the cultures. Finally, the cultures are moved to the growth room. Extensive shelving, uniform fluorescent lighting, regulated temperatures, and filters to remove airborne contaminants ensure rapid and healthy growth of the cultures. Adjacent to the laboratory, an office provides easy access to records, vitally important to the business of tissue culture. And usually, a greenhouse or a shade house is conveniently located so that newly cultured plantlets can be established in plugs or tubes and hardened off for shipping or sale. The lab setup allows for the smooth flow of people, cultures, media, and glassware from one area to another. Most critical is the movement of media from the preparation area to the transfer room and on to the growth room, and then to the greenhouse or shade house. Needless contamination of the cultures and media can be prevented if the lab is entered through a hallway, series of doors, or the office, to minimize infestations by airborne microorganisms. Now that you're acquainted with the basic layout and equipment used in a tissue culture laboratory, let's take a brief look at the 4 stages of the tissue culture process: initiation, proliferation, pre-transplant, and establishment. In stage 1, the culture is initiated. Explants taken from the mother stock are cut, disinfected, and moved to a transfer chamber for insertion onto sterile nutrient media. The tubes are capped and placed in the growth room for 2 to 3 weeks. When cultures reach sufficient size, they enter stage 2, the proliferation stage. At this time, the plantlets are removed from the tubes and placed in larger containers. With a scalpel, each plantlet is divided into small sections and replanted onto a multiplication media. Then the containers are once again returned to the growth room for multiplication. Here the cultures remain for many months, during which time the plants increase in number 2 to 3 times every 3 to 4 weeks. They must be divided and transferred to fresh media regularly during stage 2. After the last divisions are taken, the pre-transplant stage begins. The goal of stage 3 is to harden the plants for conditions outside the laboratory. This is done by using a nutrient media designed to stop multiplication and, in some cases, initiate roots. At this stage, the intensity of light striking the plants is increased. Until stage 3, the plants haven't manufactured their own food through photosynthesis. Instead, the nutrient media has been providing necessary carbon and energy for growth. After approximately 3 weeks in stage 3, the plants enter the 4th and final stage of tissue culture, the establishment stage. In this final stage, the plants are transplanted into growing media and exposed to greenhouse conditions similar to conventional vegetative propagation, but with higher humidity and reduced light intensity than is found in a typical greenhouse. This adjustment stage may last 1 to 6 weeks. We've just seen the 4 stages of tissue culture. In the initiation stage, explants are taken from the mother stock and established on a sterile nutrient media. In the proliferation stage, the plantlets multiply and are divided and placed in a larger container. In the pre-transplant stage, the plants are hardened off for conditions outside the laboratory. And in the establishment stage, the plants are transplanted and established in a greenhouse or nursery environment. On the one hand, tissue culture offers many advantages over traditional plant propagation. Space, labor, and maintenance needs are dramatically lessened, and vast quantities of pathogen-free plants can be produced in a relatively short period of time with limited mother stock. The procedure isn't affected by the changing seasons because work is done on the cellular level, not with the whole organism. Tissue culture offers a time-saving alternative to conventional cross-breeding programs and makes combinations of totally unrelated plants possible. It's also an efficient means by which genetic material can be stored for future study. But there are stumbling blocks to preventing the more widespread use of tissue culture. It's evident that startup costs for a facility and equipment can be substantial. Also, skilled labor is needed to ensure profitable results. Plants produced by tissue culture are initially quite small and must be given time to acclimate to conditions outside the lab. Any mistake in any stage of the procedure can cause contamination or genetic aberrations, ruining the crop. If you were to poll commercial tissue culture labs worldwide, you'd discover that these agricultural pioneers anticipate a promising future. Tissue culture offers an efficient and economical solution to some of the major challenges facing all areas of agriculture in the 21st century.